Hello, welcome back. Uh, Apologies for yesterday. I said the day after yesterday, the day before yesterday, that I'll be taking class tomorrow. Yeah. So what happened yesterday was I got a chance to visit. It's like kind of, you can say, an internship thing, but I was able to go and observe a surgical procedure. I mean, surgery. So I was interested in it to such an extent that I didn't even you can say noticed at the time at that place. So basically there was a surgical procedure in which there were implants, yeah. So I was so interested in observing that I forgot about the time, and when I returned, uh it was already over, and after that I wasn't able to do the drive. Yeah. Also, the thing is, I know taking a 1 day gap won't really matter much nowadays because uh I usually have 0 viewers, and nobody's really watching the live nowadays because I mean it's start, yeah. And there are certain things that I mean I have done A levels and stuff, yeah. The reason that I'm making this kind of videos is when I was doing it, I had to go through different types of resources and stuff, different kinds of things to understand the main concepts, the key concepts, yeah. So I wish that I had someone at that time to explain those things. So I want to create videos that I wanted at that time. So I mean, anybody who's doing that could benefit from it. So the thing is, even though I don't have viewers right now, even though I don't have anyone watching, but still for the you can say for the record, I just want to record this, even if in future somebody is going through these O and A levels, or you can say MCAT and stuff, and they want to understand specific chapters, so they can just go through these videos. So I'm just gonna be leaving that public, yeah. And one or two things that I believe is that yes, there are some problems. I usually didn't used to make these things on camera while just the camera on, webcam on. So I had a bit of confidence crisis, yeah. But it's improving a bit. And the second thing is my language, I think, because English is not my first language and my mother tongue, so I had a bit difficulties in usually giving the lectures, yeah. So sometimes it's like I know what I want to say, but I am not clicking the vocab or the word that should be used. So this happens sometimes, but it will just get better, I think, when I am done with, like, for example, 50 to 60 videos. But until then, I think you should just bear that. Anyways, let's just continue. So we did the introduction of cells in the first class, then we covered the basic structure of cell, then we covered the mitochondria. Wait, hold on, let me just share my screen here. So in the first class we talked about cell and its structure, the differences between prokaryotic cell and eukaryotic cell. We talked about the cell, its components, its labeled diagram. Then we proceed talking about the organelles. There are different structures present in a eukaryotic cell. So we were done with mitochondria, we were done with chloroplast, we were done with the nucleus, we were done with the ribosomes. And for the ribosome thing, I explained the concepts that were required for this chapter, but I didn't explain the translation, transcription, and things related to DNA, which I said that I'll explain it in the chapter which is after the next chapter. So we did completed the centriole part. Now what we're gonna do is we're gonna go towards another organelle, which is one of the most important one here for the synthesis of protein. So remember when we were discussing about the ribosomes, I told you that in eukaryotes most of the ribosomes are present attached to the structure, to a structure, yeah, and that structure was this one, this structure I'm talking about, this one. So a lot of the ribosomes are attached to this structure, and this is called endoplasmic reticulum. So our next organelle which we are going to be discussing is endoplasmic reticulum. Now there are two different types of endoplasmic reticulum, or in short I can say ER. So ER, so there are two different types, smooth and rough. Smooth ER and rough ER. So the key difference between these two is that on the surface of rough endoplasmic reticulum or rough ER, there are ribosomes attached. Whereas with the smooth ER, there is no ribosomes attached. Now, let me just take out the structure of it and place it here. Now, we can also represent them as these. There's two different things. So, this is rough ER, and usually the representation and visualization of this rough endoplasmic reticulum is that it contains a lot of dots, which represents the you can say ribosomes, and this one is called smooth endoplasmic reticulum. Hold on, let me just go for it and write it down. So there is rough ER and there is smooth ER. This part is usually required to remember in the CIEs or A levels, you can say. So this is called cisternae. This is the name of these structures. You can say the loops inside, or the compartment that is made due to the enfolding of membrane. So this is a single membrane structure. And the ribosomes that are attached on its surface are ATS ribosomes. So the membrane that it is made up of is selectively permeable. So selectively permeable membrane, which are folded to form like tubules interconnected with each other. Now, this part of interconnection between each other, it's the difference between endoplasmic reticulum and another organelle that we're going to talk about. The structure is kind of same, but one of the differences between them is that endoplasmic reticulum are connected with each other. The lumens of endoplasmic reticulum, which is called, the lumen is called cisternae, so they are interconnected with each other. Now, let me just explain what lumen means. This might help you in the structure of transport. So this is, for example, a vein. This is a cross section of a vein. So the inner part, this one, where there is flow of liquid, the hollow part, is called the lumen in this case. So in that case, the infolding leads to formation of a space where there are protein processed and etc. that we're going to be talking in a bit, but the processes that occurs are here in the lumen, which is called cisternae. Now, this is a very vast structure. By that I mean that endoplasmic reticulum is near, or you can say forms near, nuclear membrane, or you can say nucleus, and extends towards the plasma membrane, which is cell membrane. Also, what is the function of that? Now, the function of RER is that it is used in processing, assembling, packing and transport of the protein. Now, how does this occurs? Yeah. So let me just take this, for example, draw a structure, and this is what I understood after a long time. How does this work? So for example, this is a rough endoplasmic reticulum, and I am going to build these ribosomes that are attached to it. And for example, there are a lot of ribosomes attached, but for this specific case, I am going to build it like this. So what happens is that these ribosomes, I will tell you in the future that how protein is made using DNA and nucleotides via translation and transcription, but you just need to know that, for example, the protein is made by ribosomes. Yeah, so ribosomes produce protein strands and it goes inside the endoplasmic reticulum. Now, how does they produce it, synthesize it? This is none of your business right now. Yeah, you don't need to worry about that. But now the strands of protein or protein chains, polypeptide chains, are thrown into the cisternae of the endoplasmic reticulum, or rough endoplasmic reticulum for precision. Anyways, these simple structures, secondary structures, remember we talked about this in the chapter of biological molecules, the primary, secondary, tertiary and quaternary structures. So by the processing means that this specific structure, this structure is gonna be processed. Now this is in, for example, secondary form. This protein is in secondary form. Now inside the rough endoplasmic reticulum, this protein is gonna start forming bonds because they provide a specific environment for proteins, and this is what we call processing. Now after the part where there is processing, the proteins are gonna be converted into the tertiary structure or functional structure. So before they were in the form of secondary structure, they were straight chains, straight polypeptides, and there were no use at that point. But to convert them into functional part, the rough endoplasmic reticulum processes them. Now, for example, this is a globular protein processed inside the rough endoplasmic reticulum. Now, assembly. What does we mean by assembly? Now, this is then gonna travel to, let's say, here. Now it's gonna travel to, let's say, here. So it travels to this part, and now we're gonna travel it and accumulates inside this. So this is what assembly means. It assembles the necessary specific proteins, assembles inside the lumen at a specific place. And then what happens is the next thing that we wrote here is packing. So what does packing means here? So these membranes, after the required proteins have assembled, starts to shrink, starts to pinch, and eventually this part here breaks. So what happens is, after being pinched, this part breaks and it forms a vesicle, and this vesicle is then transported. So this contains protein, assembled protein that is required. This is pinched off, and the pinched structure that is pinched off, budded off, is now in form of a vesicle, and this vesicle is made up of single membrane, and this contains the proteins that are required. Now this structure then further completes itself and form another vesicle here. So this process occurs a lot of times, that protein assembles here, then it pinches off, and a new vesicle is produced, and it goes further on and on and on. Yeah. So this is what happens. And this vesicle containing the protein can go anywhere. And by anywhere, I mean it can move towards Golgi body. It can move towards towards other cellular components. It can move extracellular outside the cell for some functions. For example, let's say insulin, for example, hormones. They're produced inside cells, but they are used outside cells. Intercellular, they are intercellular proteins. So some are intracellular, which are used inside cell, and some are intercellular that are used outside of cell. Maybe let me just repeat that. Intercellular are the ones that are used outside of the cells, and intracellular are the ones that are used inside of the cells. So let's just keep this vesicle here. So as I told you, this vesicle contains the globular form of protein, and this is rough endoplasmic reticulum. We will talk about where it goes and what happens to it, because the next organelle that we're going to be talking about is involved with the processing of this specific vesicle. So we're going to just keep it. Go and wait there for your turn. Anyways, there is another type of endoplasmic reticulum, which is smooth endoplasmic reticulum. And yeah, so smooth endoplasmic reticulum is called SER, and its chief function is to synthesize lipids. Another function that is not usually taught in A levels, but I used to know about it because there was an MCQ related to it. So there's another function of SER, which is storage of the you can say ions. So for example, sarcoplasmic reticulum, which is present in muscle cells, which releases calcium ions, is a kind of smooth endoplasmic reticulum. So I'm just gonna write it. We're gonna be talking about this specific part in the chapter of support and movement. So the muscular anatomy and stuff will be discussed at that point. But for the reference here, I'm just gonna write it. So uh so there is a name of vesicle which is called residue, you can say. Anyways, let's just talk about another major component or major organelle here. So but wait a second, let me just drink water here. Now let's just go towards another structure here. So where did we left our vesicle? So this is our vesicle that was formed from rough endoplasmic reticulum containing a tertiary structure of protein here. Yeah. So now this travels to another organelle, which is here. This one. So this structure is called Golgi body. There was an error, I think. Anyways. So the lumen of this specific organelle is also called cisternae. But there is a difference between you can say ER and the Golgi body. So Golgi body was discovered by Camillo Golgi. I don't remember the year here, but he was an Italian scientist and from University di Pavia, I think. Anyways, so there is a thing that you should remember with regards to Golgi body, which is the cis side and the trans side. And I'm gonna explain it in a bit, but first we need to discuss what is the function of it. So, for example, where was the vesicle that we left? Yeah, so here is a vesicle that came from rough endoplasmic reticulum. Let's just make it small, reduce the size. So this vesicle contains proteins. It comes towards here and attaches with the Golgi body. So let's just, so it comes here and attaches with the Golgi body here. Now what happens is the membrane infuses and the protein present inside the vesicle goes inside the Golgi body. So this happens. So first the membrane was invaginated and budded off in form of protein, and after that that vesicle travels towards the Golgi body if the protein required needs to be modified. Yeah, so it travels towards Golgi body, attaches itself, infuses its membrane, and releases the protein that is present inside it to the Golgi body. Now let's say, for example, there is another vesicle that arrives here and leaves its protein here. Now what's going to happen is the proteins that needs to be required, for example, inside the body, there are some proteins that are glycoproteins, there are some proteins that are lipoproteins, there are some nucleoproteins. So the proteins that involves another compound or the conjugate molecules, for instance, for example, our cell membrane includes the glycoprotein, which is carbohydrates plus protein. So where is those gonna formed? So they are gonna form inside the Golgi body. These specific modifications with the proteins, for example, this structure is in form of tertiary structure, and when it needs to be in quaternary form, it's going to be modified inside the Golgi body, where some iron is going to attach to it. Anyways, for example, in the hemoglobin molecule, where there is the quaternary structure, 4 chains, alpha, beta, 2 alpha and 2 beta chains of proteins are attached with each other. Similarly, if the protein needs to be in the quaternary form, where, for example, these are 2 chains, they needs to be attached. So they're not going to attach themselves inside the rough endoplasmic reticulum, but they're going to attach themselves inside the Golgi body. So this is the key difference that you need to understand here. Golgi body are not interconnected tubes. They are not interconnected tubes like rough endoplasmic reticulum. This is difference number 1. Also, rough endoplasmic reticulum is used for the 4 things that I told you: transportation. Let me just brought that up. So they're used in processing. The synthesis of protein is inside the ribosome only. That ribosome transfers the protein chains inside the rough endoplasmic reticulum. Those rough endoplasmic reticulum processes, assembles, packs, and transport the protein. Now this transportation can be outside cell. If not, that protein needs to be modified in modified form, because it's useful in modified form. It is required to the cell in modified form. Then it goes towards the Golgi body, and after it reaches to the Golgi body processes it, modifies it, and transport it to the areas that it is required. Now what are those modifications? For example, transforming the tertiary protein into the quaternary structure, transforming the protein into conjugate molecules, for example, glycoproteins, lipoproteins, etc. This is all done by this is all done by the Golgi apparatus, Golgi body. Now let's just list down the points here. So it's the series of folded membrane. This is a single membrane structure. You need to remember this, as I've listed all the single membrane and double membrane structures in the starting of this chapter. It is a series of folded membrane arranged in a group. Just group of 4 to 7 individual stack structure. These stacks are flattened sacs and are constantly forming its one end of Golgi body from vesicle, which buds off from ER, and another side modified protein is budded off from the Golgi body and transported by Golgi vesicle. So what does this mean? So continuously what is happening here is a vesicle is coming from the rough endoplasmic reticulum, or you can say endoplasmic reticulum in general. A vesicle is coming, and remember I told you in smooth endoplasmic reticulum lipids are synthesized. So these, for example, you want to make lipoprotein, so from the smooth endoplasmic reticulum a lipid is going to come. From the rough endoplasmic protein is going to come. These vesicles are going to form conjugate molecule inside Golgi body. They're going to be modified, and then they're going to be accumulated at the side or the end side of the Golgi body. And then what's going to happen is that the membrane invaginates. And how does this happen? Let me just give you a brief example here. So this is a membrane before invagination. Or let me just, so for example, this is the end of Golgi body. Now what happens is this starts to short from specific side, which is invagination of membrane, pinching off of membrane. So what happens is over a bit after time, this membrane is going to be like that, and from the top it's going to be like that. And after some time, they're gonna, so there is a protein here and the invagination is occurring. So after some time the membrane is going to be like that, and after a lot of time, you can say the time when invagination occurs and there's time of budding off. So what happens is the wall is folded enough, for example, the wall is folded enough that it breaks off, and a Golgi vesicle is formed which contains the modified protein, which is ready to be used. So I think I've explained enough of the Golgi body. Let's just move towards the points that you need to, the notes, for example. So the stacks are flattened sacs. So from one side it infuses itself with the vesicle from endoplasmic reticulum, and from the other side it releases or buds off the Golgi vesicle. So the main function, as I've explained, of Golgi body is to modify So by the modification, as I've explained, they can remove unnecessary amino acids, join chains of amino acids, other amino acids, they can modify by adding other molecules. Performing conjugate molecules. Bear with me writing, it's very bad. I'm sorry for that. That's why I'm just repeating everything. So forming conjugate molecules, e.g., for example, lipoprotein. So Golgi body modifies the processed protein from the endoplasmic reticulum. Also, they may remove some unnecessary amino acids or join or add different kind of chains or amino acids which can form quaternary structure, can modify by adding other kinds of biomolecules apart from the protein. So from other molecules, I mean lipids, carbohydrates, etc., for forming conjugate molecules, for example, lipoprotein. Yeah. And they transport it as well, and which is called the Golgi vesicle. So it buds off from here and in form of Golgi vesicle. And there are different types of vesicles. Some kinds we're going to be talking about, for example, in cell there are different kinds, for example, peroxisome, glyoxysomes, lysosomes, and these are different kinds which contain different kinds of protein and stuff. We're going to talk about it. And these usually, a lot of them are formed from the Golgi body. Now, one more difference that we have here is, and this is a technical thing here, Golgi apparatus does not contains any nucleic acid. Now you're going to think, if we're going to compare it with the mitochondria or say chloroplast, then it makes sense to mention here that it does not contain any kind of nucleic acid. But the comparison was between endoplasmic reticulum and the Golgi body. Now, why we need to mention this? So we need to mention this because rough endoplasmic reticulum, yes, it contains nucleic acid, and the technicality behind it is because ribosomes, as we have discussed before, are made up of rRNA, which is ribosomal RNA, yeah, nucleic acid, ribonucleic acid in full form. So ribosomal RNA is present in the ribosomes, and while ribosomes are attached to the rough endoplasmic reticulum, they are considered a part of rough endoplasmic reticulum. That's why there are two different types of endoplasmic reticulum, rough and smooth. So the difference between them was the attachment of the ribosomes, and because rough endoplasmic reticulum is considered as a combination of you can say ribosomes and endoplasmic reticulum, that's why we can say technically that rough endoplasmic reticulum contains a nucleic acid in itself, not inside the structure, inside the lumen, but on the surface because of the presence of the ribosomes, because ribosomes are made up of RNA, which is ribonucleic acids, whereas Golgi body does not contain any kind of the ribosomes attached to it, so they are not gonna be called, or you can say that they does not contain any kind of nucleic acid in their structure. So there is a thing that I told you in the starting here, which is the cis side and the trans side. So vesicle from endoplasmic reticulum arrives at the cis side of Golgi body, and vesicle attaches itself to the cis side, and the membrane between Golgi body and vesicles are you can say dissociated, or you can say invagination. Due to the invagination of membrane, they are the Golgi vesicle that forms. The formation it is at trans site. So after content being released in cisternae, where it is modified, and after the modification, invagination occurs and vesicle is broken or bud off from the trans side. For example, there is the attachment or infusion of the membranes of vesicle from ER and the Golgi body. So this is going to be called cis side, and after that, let's just remove it. After that, this transfer from here to here, then from here to here, and for example it buds off from here and the vesicle goes in there. So this is gonna be called trans site, because from this side the Golgi vesicle forms, it's bud off, the membrane is you can say broken at this side, whereas it was fused within the membrane. This side was called cis side, the site of infusion, and the side of breaking or budding off is called the trans site. So let me just ask you one question: which of the side contains the modified protein? And the answer is going to be the trans site, because after several modification, when the protein, or you can say the modified form, is needed to be transported, it is sent towards the trans site for being budded off, whereas the side which does not contain the modified protein is going to be the cis side, because it's the infusion of the membrane. Anyways, let me just draw a pathway for the synthesis of protein. So from ribosomes, either from the cytosol or you can say attached to the endoplasmic reticulum, protein is made and sent to ER. Then it is processed in ER, or specifically RER. It is processed there and it is sent towards Golgi body. or apparatus, and then it is sent to vesicles in form of secretory vesicles, or you can simply say vesicles. And via vesicles it goes in and out of cells, so it can be used intracellular or intracellular. So if I draw a diagram, but it's gonna take a lot of time, so I'm just gonna draw a diagram in tomorrow's class before even starting the class. So I'm just gonna paste that diagram for your better understanding how protein synthesis works. But you need to remember the order here, yeah? So if, for example, we say in this diagram, let's just go towards the cell diagram. So there's a question, for example, they ask you label the diagrams where the protein synthesis occurs. So they are 4 options. They, for example, give you 4 numbers, 3 and 4. And they ask which are the 4 steps where the protein are processed. By which 4 processes does the protein, you can say processes. So you're going to label it. So the first one is going to be towards the ribosome. Then the second one should be the endoplasmic reticulum. The third one should be the Golgi body. And the fourth one would be vesicles. Got it? Now, let's just move towards another organelle. But before that, let's just mark of the organelles that we have done. We're done with endoplasmic reticulum, and we'll do lysosomes are left, cytoplasm is left. Now, let's just talk about cytoplasm. Cytoplasm should have been covered first, but I don't know why I didn't cover it in the first place. But anyways, cytoplasm. Just drink a bit of water here. I think we should end this chapter today. It's gonna take 30 to 40 minutes more, I think. Let's see, anyways. So there is a word which is called protoplasm. You might have heard about this word. What does this word mean? So protoplasm is the living content inside the cell. One thing I should tell you is that the cell wall is considered a non-living content. But anyways, the living content of the cell is called protoplasm, and you can say the alive content which is required in the reactions inside the cell, or you can say involved in the spontaneous reactions, which is the part which is very important inside the cell, and without it life will cease to exist. That part inside the cell is called protoplasm, and without protoplasm life will not exist. Yeah, so there are two parts, you can say, that are included in the protoplasm. One is nucleus and cytoplasm. So these two things forms up the protoplasm. By cytoplasm, I would highly be involved. There's likely chance that I involve mitochondria, all the organelles. I know nucleus is also a part of cytoplasm, but it isn't required to mention it separately, because there are some functions that are excluded in the reactions. For example, nucleus does not carry out the chemical reactions that are included or that are required for the cell's survival, but it is the thing that is, for example, used to how should I say this? Hold on. So nucleus contains the set of instructions for specific reaction to occur, and the cytoplasm is the place where those reactions occur. Got it? So for example, nucleus, I can't really explain how does it happen because I have not explained you guys about the transcription and translation, but for instance, I'm just going to tell you that nucleus is involved in the replication, the reproduction of cells. It is involved in the synthesis of protein primarily, or you can say the instructions that are required for a specific reaction to occur. So nucleus is a thing that is the boss here. That is the you can say leader, and cytoplasm are the workers that work and do chemical reactions for the survival of the cell. Cytoplasm contains those components which can be mitochondria, chloroplast, ribosomes, lysosomes, endoplasmic reticulums, Golgi bodies, glyoxysomes, peroxisomes, etc. You name it. And cytoplasm contains these kind of things. Every cytoplasmic content is important for the life. And in the nucleus, they both form the protoplasm here. So what cytoplasm is? So cytoplasm is a aqueous substance, or you can say water-based, because most of the percentage that is made up of is aqueous, or you can say water. So it contains different organelles and cell structures and substances like the waste products and the storage. Now, uh, wait, hold on, my screen. Sorry, I noticed that. So, the difference. So, cytoplasm is divided into two things. Yeah. As I told you, one contains the aqueous substance, and the one contains different organelles. So the part of cytoplasm which is usually the soluble part, the water-based part outside the organelles, is called cytosol, where most of the reactions occur. Yeah, so the reactions like glycolysis, cell transduction, signal transduction, and these kind of things occurs in the cytosol part. Now let me just explain you with the diagram, with the help of diagram. You might understand this quickly if I do this. Let me just paste the structure of cell here. So for example, this is a cell. Now I'm gonna highlight what is the cytoplasm here. So let me just change the color. Let's say purple. Let's just take the blue. So all of the blue part here that I'm gonna be marking is the part of cytoplasm. So all of the part that I have marked here with the blue is what cytoplasm is. This is all what cytoplasm is. Now there are two types of, there are two parts of cytoplasm. One is this part that is empty space that contains soluble substances, yeah? And another one is this part, this part, this part, or you can say this part, this part, this part, this part, this part. These are all parts. They are, though, the part of cytoplasm, but they are not soluble. They are different compartments. So the part of compartments, the area of compartments is cytoplasm, but the area, though being the part of cytoplasm, the part where there is soluble substance, the gap you can see, the empty spaces where most of the reactions occur, is called the cytosol. One thing more, cytoplasm also contains cytoskeleton, which is made up of very fine fibers of microtubules. So let me just again draw the structure of cell here. So for example, this is a structure of cell. Now I should be drawing a bit of what cytoskeleton looks like. So one thing I want to clear out is I have though drawn these passings, but they does not usually, or they does not pass through the organelles, but they are between the organelles. Organelles are between the cytoskeletons, the microtubules, but they're the main function of these cytoskeletons, the presence of these cytoskeletons, is they assists in the movement of organelles. For example, there is less light here and there is more light here. Let me just remove. For example, this part does not contain sunlight. Or let me just remove this. This part does not contains sunlight, but this part contains sunlight. So sunlight is coming at this part. So now what's going to happen is, because chloroplast was present at this area, cytoskeleton would assist them to move from here to here. So for the movement of the organelles, the systematic movement, cytoskeleton is the reason which assists them for that movement. And also, one thing more, if there was no cytoskeleton, because of the soluble part, the cytosol, the water-based thing, the organelles would have been just floating here and there without any coordination. It would have been just random, you can say, movement of organelles. And you can say if a gene comes out of the nucleus, it would have been just roaming here and there with the flow of the cytoplasm. But the systematic flow of gene getting out and then moving to the area where it is required, or lysosome moving out and going where it is required, this is due to the systematic movement with the help of cytoskeleton, which is made up of the microtubules. Got it? No. It contains 90% of water and forms a solution which contains fundamental biochemicals of life. So large molecules such as protein that are present in cytoplasm forms a colloidal solution. Cytoplasm can be both a runny solution or a gel-like solution, which means that it can be concentrated and it can be, you can say, what is the opposite of concentrated, bro? I just forgot the word. Anyways, you get what I'm trying to say. Most probably the outer part of cytoplasm is jelly-like. I mean, it is more concentrated, saturated, and the inner part, the ground substance, or the inner part, is more like a runny, or you can say unsaturated, less saturated solution. And the ground substance is the site where metabolic pathways like glycolysis occurs. So I think these points are enough for cytoplasm, as also a lot of times I don't write the points, but I explain them. So if there's something that you get out of those points, there are, for example, some points that I explained about cytosol, and there are some points that I explained about the reactions, and during the explanation with diagrams I usually give some points, which if you think that it is important, you can write it, but I usually write the points which are the highlights of that you can say topic, but you can write a lot of points from the explanations that I give. So it's better to extract out points, and the main thing is concept here. If you get the concept, a lot of MCQs are going to be easy for you. We're going to be solving the MCQs too. After the completion of these 2 chapters, we're going to be dedicating 2 classes for specifically for MCQs. Anyways, I think the class is 1 hour and 14 minutes now. So I said that we should be completing this chapter today, but I think we should just end it tomorrow, because there are not many things left. But first I need to just write one more point here that I think is important here, that I should have mentioned or wrote, which is about cytoskeleton. So cytoskeleton is a structure that helps cell maintain their shape, or provides mechanical support, and for help in transportation, inside cell. So what we are left with is, we have done the cytoplasm, and what we are left with is the cell wall and the lysosomes and some 2 to 3 organelles, which we're going to be just going through quickly, which are glycosomes and stuff. So we're going to be winding up this chapter tomorrow, and I think we would start the chapter of cell membrane in the class after tomorrow. After completing this chapter tomorrow, I think it will be in 30 minutes. After that, we're going to be starting the cell membrane, or you can say the membranes part. And after that, we're going to be going towards genetics, or you can say cell reproduction. So it was enough for today. I hope you enjoyed it. I hope your concepts are clear. Some of your concepts are clear. If some of the things were left unclear, you can comment. I would reply definitely, and cover them or explain them in detail in the next class. Until then, I would say bye bye, take care, see you in the next one.